Good afternoon, Dr. Gustav. Good afternoon, class. Today, Group 5 will be presenting on the topic, the bioethical implications of gene editing. Our group members are myself, Sharon Cornish, Trayvon Wood, Jade Russell, Alexis Williams, Sierra Sayers, Crystal Clark, Amatravia McPhee. What is gene editing? Gene editing or genome editing is a method that lets scientists change the DNA of many organisms, including plants, bacteria, and animals. Every cell in our body contains a copy of our genome, over 20,000 genes, and over 3 billion letters of DNA. Our genes shape who we are as individuals and as species. Gene editing has been everywhere on the news in the past few years and has been capturing the scientific media. A revolutionary new gene editing technology called CRISPR has now been developed that significantly improves the process of singling out pieces of DNA and replacing them. This is made up of enzymes called Cas, which acts as a pair of scissors to remove the sequence and to insert a new one. This is a group of technologies that enable scientists to change the DNA of an organism. These innovators permit genetic material to be added, removed, or altered at a specific location in the genome. The genes are sections of long DNA molecules coiled up inside each cell of every living thing, from microorganisms and insects to plants, animals, and humans. Alongside some ethical beliefs, benefits of gene editing, there are also some ethical and social concerns to consider. First, one type of gene editing is the somatic gene editing. In this type of editing, you take the consent patient cell and you perform gene editing on the patient. For example, someone who has hemophilia, there is a replacement of pieces of DNA. The Cas9 allows a patient body to produce their own blood clotting factor. It uses a piece of genetic material and an enzyme that acts like molecular scissors to repair the genetic fault that causes clotting factor deficiency. In another type of gene editing called germline gene editing, this type is the same concept, but the embryonic changes goes from generation to generation. For example, breast cancer. Using a Cas9, specific loss of function or gain of function mutations could be achieved. So, the gene could be removed to make daughters and granddaughters free from breast cancer or to minimize risk. In another example, there is a disease in the Eastern Europe and Africa, swine flu, which a lot of people die from this infection. The warthog is resistant to this infection, so one thing which has been done in domestic pig is to take part of the genome of the warthog and change the gene with the RELA gene in the domestic pig, which renders the domestic pig with resistance from this form of flu. In embryonic gene rearrangement, the public supports such cases as disease prevention, such as hemophilia, cystic fibrosis, Huntington's disease, and sickle cells, and less support in enhancements such as increasing height, changing the eye color of a human, or increasing more hair or hair growth. Technology is already in our hands. Now it's time to debate on how we use it, regulate it, avoid negative uses, and unlock its potential to provide life-changing solutions to some of the world's biggest challenges.
The definition of gene editing is making a small controlled change in the organism's DNA. This small change in the DNA structure can turn into a major change in the organism. This will create genetically modified organisms or GMOs. The most common way to produce GMOs is by CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats. CRISPR-Cas9 is the most common genome editor because it's an RNA-based system. It works by cutting a DNA sequence at a genetic location, then deletes and replaces the DNA sequence. Gene editing is very beneficial and useful for plants and animals. CRISPR allows scientists to edit specific DNA to enhance beneficial characteristics of plants. Plants become more nutritious and grow at a faster rate than before it was genetically modified, which helps for the high demand for edible plants. The process for improving plants is better than any form of plant gene editing and leads to healthier and stronger plants. Animals are less susceptible to sickness and diseases. They are overall healthier without gene editing. They grow and mature faster. Also, they produce more offspring during the mating season. Gene editing is important to the agriculture industry. Plants in agriculture are more resilient to climate change and tougher climates, and are biologically designed with gene editing, able to withstand against droughts, irregular weather patterns, and pests. Gene editing helps to fight against genetic human diseases by sniping out diseases causing mutations. This is known as gene therapy, which involves altering the genes inside your body cells in an effort to treat and stop diseases. Gene therapy replaces a faulty gene or adds a new gene in an attempt to cure the disease or help the body's ability to fight diseases. Gene therapy can treat against many diseases such as cystic fibrosis, cancer, heart disease, and diabetes. Gene editing makes the world a better place for living things, whether it be humans, animals, and plants.
There are many different benefits when it comes to the use of gene editing. One such benefit is the curing of diseases. Gene editing is now being used to treat diseases by changing the correct gene that causes a disease. As of right now, researchers are looking to see if gene editing could be used in the fight against cancer. Researchers wanted to see if by modifying the genes, the cells of the immune system can be enhanced, resulting in the cells being able to better detect and kill cancer. At the moment, only 3 tests have been conducted so far on 3 different willing patients. In 2 of the patients, their cancer cells had their production stopped after gene editing was used, but only for a brief period of time. In the 3rd patient, however, there was no change. Although the progress may seem small, the fact that any change at all was made means that there is a promise in gene editing being used to stop cancer. Another benefit of gene editing is to help save endangered species. Gene editing can also be used on animals and plants and can help to benefit many species that are seeing a rapid decline. Some animals are on their way to becoming extinct not only because of human encroachment, but also due to changes in climate, other invasive species, and diseases that only affect a specific animal. One such animal that is affected by this disease is the Tasmanian devil, which suffers from a facial cancer that has killed over 80% of the species population. Not only would gene editing allow for us to save the species from dying out, but it can also give us more insight on how to combat cancer within humans as well. Scientists have discovered a protein in the Tasmanian devil that stops the immune system from creating an anti-cancer response. This protein in the Tasmanian devil has also been found in humans. From this discovery, scientists have been trying to find a way to use gene editing to either boost the immune system, get rid of the protein, or to create a vaccine that can protect the Tasmanian devil from this cancer. If this proves successful, this can also be the blueprint of creating a cancer-preventing vaccine for humans as well. Another benefit to gene editing is the increase in food production. Gene editing can design foods that can withstand harsh temperatures and can hold more nutrients. Crops can be made to produce more yields and can also be used in a way that it will take less materials to grow certain crops, meaning that food prices would go down as farmers would have to use less materials in order to make crops grow. This can benefit us greatly as this can also help to meet the high food demands that have yet to be met in certain countries. Moral theory in favor of gene editing is beneficence. According to Savulescu, using gene editing in the fight against diseases align with beneficence, which is the moral obligation to benefit people. Gene editing would benefit people greatly as it would not only stop the diseases that would otherwise cause people pain, but it would benefit them by having gene editing as it would give them a chance at a better life and to no longer experience this discomfort. Gene editing is a good step in the right direction on how we can maximize happiness and how we can benefit each other by helping to stop diseases, increase food production, and also help species that may see themselves endangered.
Pest-resistant crops is another pro of gene editing. When we think of farming and food production, one of the challenges persons face in the industry is the infestation of their crops that can happen through insects and any harmful kind of substances that is surrounded by it. So by using this form of genetic engineering, it allows for plants to be cultivated with their own built-in pest repellent, so they're able to protect themselves against any kind of insects or pests that may be harmful to them. Some examples of these insects may be the cotton bollworm or the European corn borer. From a utilitarianism aspect, I do believe that pest-resistant crops is very beneficial. When you think about it, it's almost like a domino effect, because once these crops are able to protect themselves in a sort of way, then they're able to flourish. And once they're able to flourish, then farmers or whoever are able to produce them on a larger scale and distribute them to more persons. So it helps the majority to get these crops or anything that is being grown and to have it in a way that it's in a nice quantity but also in a good quality as well. So moving on to novel medical treatments, these kind of treatments are used to help to cure illnesses, but also helps to introduce an innovative way of treatments into medical care. So as we see science progressing and just technology making advancements daily, there are going to be times where we see novel medical treatments coming out because we have these things that are happening in research and just in the medical field. So through the use of novel medical treatments, it would help to aid in creating effective more resources for patients and just persons in general who may have an illness or a need in seek of treatment. That some may argue that practicing gene editing or even any form of genetic engineering is a risk. But if we look at the AstraZeneca vaccine that we've been using during the pandemic, that was also a form of genetic modification. So it's not like this practice or this usage is new in the medical field. However, I think that if we start to use it more, it would be very beneficial because it would help us to just create new treatments that can be very effective. Lastly, another pro of gene editing is the increase in life expectancy. So because patients are able to get the treatment they need through gene modification, this aids in them getting better and having a better quality of life, and also improves their health and their state of well-being. Also, to this statement or this kind of argument can be used to say that it helps to preserve mankind as well, because this all goes with just helping to improve our well-being and just preserving us so we can be here for a long time.
During the gene editing process, there's a high chance of making mistakes. Errors have the potential to be harmful. A researcher, for example, may inadvertently delete a gene. The fetus may acquire developmental problems as a result of this. Any mistakes in the germline editing could be passed down through generations. A lab test showed risk of using CRISPR gene editing on embryos. A lab experiment aimed at repairing defective DNA in human embryos highlights what may go wrong with gene editing and why scientists are against it. The editing resulted in unexpected modifications in more than half of the cases, such as the loss of an entire chromosome or large portions of it. Several panels of scientists and ethicists have stated that it's too early to say if this can be done safely, and the latest experimental data showed potential danger. Lastly, safety concerns. Since scientists are still experimenting with gene editing, they are constantly experiencing unexpected results. Moreover, complications can occur such as miscarriage, stillbirth, or premature birth. In my opinion, gene editing should be made illegal because modifying someone's genetics puts them at risk. The outcome is unknown until analysis occurs. Also, is irreversible, so if the results are harmful, the person will have to live with this outcome for the rest of their life and pass the defect down their family line.
Extremely high costs for treatment. Vaughan reasons that if there is a treatment available to cure certain diseases and save lives, shouldn't it be offered? However, if these treatments are extremely costly, who then really benefits? Only those that can afford the treatment. In a previous presentation, the question should our healthcare system in the Bahamas allow for genetic testing for elective abortion decision making was put forth. During this time, information was presented on the costs of the tests that were able to assess if a fetus had genetic disorders. Costs that were disclosed range between $900 and $950. If this is just the test to detect the defects, imagine the price to alter or repair them. Managing Director of Pharmaceuticals and Medicines, Rich Durante, explains that though genetic therapy, a form of gene editing, is exciting and shows great promise in identifying and treating rare disorders, it has the possibility to be very costly. In numerous speeches and presentations, experts expressed the hopes of gene therapy being the big and the greatest solution to the second largest cause of death in America, cancer. According to Karen Selby, as it stands, depending on the type and stage of cancer, the treatment alone can cost as much as $33,500 per dose. There is a false narrative spewed when drugs and treatments are being created that the more persons that are affected by the disease, the less the treatment will cost. However, this is noticeably misleading as cancer is the second leading cause of death among millions. If genetic testing alone is almost $1,000, what can the actual cost of receiving treatment be? Gene editing can lead to the elimination of characteristics that are deemed undesirable. As technology advances, genes that can alter basic characteristics such as gender, eye color, or even height can now be manipulated. In the article How to Buy a Daughter, author Jasmeet Sidhu shares the story of how a mother desperate to have a baby girl left Canada, where gene selection is illegal, for the United States and paid $800 for a scientist to ensure that she would birth a baby girl. The Times Online posted a story of a British couple that won the right to test embryos that would ensure that their offspring would not carry genes that caused high cholesterol or risks of heart attacks. New Scientist report of a deaf lesbian couple that chose a deaf sperm donor to ensure that their children would also be deaf. Parents have the right and the ability to choose features that are preferable to them. This is a basic form of eugenics, the deliberate attempt to improve the genetic makeup of humans by manipulating reproduction. Vaughan compares eugenics to the Nazis' drive to racial purification through mandatory sterilization of undesirables. Today, it may be eye color, tomorrow it could be skin color. This improving technology could potentially eliminate undesired races. When taken too far, gene editing can go against natural law. In 2015, ABC News reported that two scientists in Canada altered the gene in an apple that causes it to turn brown. Numerous articles state that the browning of an apple can be used to tell when the apple is rotting. Rotting apples can become the home for toxic bacteria that can potentially kill parts of the liver, cause organ failure, and suppress the immune system. By removing this gene, scientists essentially eliminate the apple's natural ability to display when it is no longer edible.
A Bahamian example in regards to gene editing and the negatives it may pose in the future is shown in the Tribune article from February 2021, when the government found that foreigners who used the genetic material from our marine resources was using a loophole and exploiting us. Hartnell, The reporter of this article stated that the former Minister of Environment, Romuald Ferreira, had proposed a legislation in regards to the permitting and revenue sharing of those using the genetic material found in our marine resources. This comes only after a report from the Inter-American Development Bank, or IDB, revealed that the Bahamas is not being compensated from the countries that were using and editing the genetic material from the Bahamas marine resources in the creation of new medications. The two main countries that were doing this were the US and Canada. The Bahamas was receiving no commercial or economic benefits from these countries whatsoever. One of the genetic materials that a pharmaceutical company used from a Bahamian microbe strain reached clinical phase 2, and the Bahamas is not compensated for this, nor were they made aware. This Bahamian example shows the negatives that gene editing poses, as the Bahamas was used as a means without being compensated and was instead exploited. Could this happen to other patients or other countries who...
The Biological Resources and Traditional Knowledge Act sets a foundation that includes the Bahamas in shared benefits that arise from the use of genetic resources, access to knowledge and technologies, and funding to protect and preserve genetic resources. In this act, persons that are not in compliance with the proper procedures concerning the retrieval or use of genetic materials are penalized accordingly. Penalties range from responsibility of repairing damages caused by unlawful disruption to retrieve genetic materials to fines up to $10 million.
My name is Crystal Clark, and I will be presenting 5 reasons why my group is against gene editing and enhancement. 1. Informed consent. Permission from the embryo for treatment. It is impossible for embryos to give informed consent. This makes the act ethically wrong. Such embryos may grow up, then be told by their parents how they came about and why they are resistant to certain diseases or conditions. It is possible that these children may get upset with their parents for giving consent for their genes to be altered, simply because they wanted to be like others instead of being gene-edited humans, which will make them feel like they are abnormal or awkwardly different from others. Respect for autonomy. Lifelong testing. Because the use of gene therapies is so risky, it will call for frequent testing. No one would enjoy having to visit the doctor frequently to have tests run just because their parents had scientists manipulate their genes. This can be depressing and unfair for those individuals that would be forced or even tricked into going to the doctor for a simple checkup just to get those tests done. This goes against the principle of respect for autonomy. Unfairness of genetic enhancement. Walter Glannon's viewpoint. His view speaks about purchasing genetic enhancement. This results to parents paying for their children to be genetically enhanced, which Walter Glannon sees as a morally wrong act. Unfair advantage. Engineered children having advantage over non-engineered children. Children are engineered to be faster, smarter, stronger, etc. These are some of the enhancements that parents may seek for their children. For example, students in school participating in track and field. If one parent paid for their child to get genetic enhancement, that makes the child stronger to get a gold medal for participating in shot put. This would be unfair to the non-engineered competitors that have their natural strength in which they are born with. This is unfair because it gives the engineered child somewhat of a head start to get the gold medal over the other children. Undeveloped and unsafe. Lack of technology. This leads to more dangerous diseases and conditions. Scientists don't have the proper tools needed to safely carry out these therapies. Genes are often linked to each other in important ways, so by manipulating or enhancing a gene can affect other genes. Negative side effects. sickle cells giving resistance to malaria. Gene therapy aims to cure sickle cell, which is good. However, by removing the gene that is responsible for sickle cell has a negative side effect. It can cause malaria cases to rise because these patients are now liable to contract malaria. Therefore, it is possible for other enhancement to have similar negative side effects. Mosaic twin girls. These are the first gene-edited human beings. Mosaicism were present in both Lulu's and Nana's embryo, also in Lulu's placenta. While it is good that the twin girls are immune to HIV, according to Yale Medicine, mosaicism is when someone has two or more sets of cells that differ genetically from one another. This leads to development of diseases, which brings about a negative side effect relating to gene editing in the twins, which is similar to the sickle cell and malaria case. Cost of gene editing procedures. Gene editing is unnecessary as it relates to HIV. HIV-positive parents giving birth to a HIV-negative child is a pre-existing privilege. Viral suppression prevents that child from contracting HIV. Viral suppression is defined as reducing the function and replication of a virus. Antiretroviral therapy is the regimen used that reduces a person's viral loads to undetectable levels. How does this work? First, both parents should check their viral loads. Then, begin to take medication. After 6 months of taking medication, then both parents' viral loads should be at the undetectable level. Then 6 months from that point, they should get another test taken showing that their viral loads remains at the undetectable level. If both tests follow along these lines, then it is safe to conceive. The mother should continue taking medication during pregnancy and while breastfeeding. This is how the child remains HIV negative. Expensive. The cost of gene editing is far more expensive than antiretroviral therapy. The cost of HIV medication ranges anywhere from $1,800 to $4,500. There is no specific cost as it relates to gene editing. However, we can estimate that it will be very expensive, taking into consideration the type of procedure. Therefore, only wealthy people would be able to benefit from this, which puts the lower and middle class at a disadvantage.